Hi everyone, we are Section 7, Group 2, and our project examines the effects of APP and Tau on fruit flies' physical structure and behavior. We are also inspired by the hypothesis that sleep decreases the APP and Tau levels. Thus, we also wanted to examine the role of sleep on decreasing or increasing the levels of these proteins. Before we actually start delving into our project, we just wanted to state that our group members were conducting the experiments online fully remotely since week 7. Therefore, our results are based entirely on our hypothesis that is inferred from credible papers that are published before. My name is Cece, and I am currently studying neuroscience and computer science at BU. I aspire to incorporate more of the principles of neuroscience into the technological industries for implementation, such as improving machine learning or image capture or developing smart housing.
Hi, my name is Sammy Merrill, and I am a senior studying neuroscience at BU. I enrolled into a STEM program in high school, which really started my love for science. I intend to work in the field of addiction research once I graduate, and I am also attending MCPHS in the fall to study nuclear medicine.
Hi, my name is Pearlie, and I'm a junior in neuroscience. Neuroscience research comes from understanding how the brain works, which yields a new perspective to each investigation. In pursuing a bachelor's degree in neuroscience, I will establish a foundation for my future academics of brain diseases and future role as a medical practitioner. So while taking an introduction class to neuroscience, the professor had presented a focus on Alzheimer's disease during lab. We all took an interest in the effect of base mutation on plaque formation in Alzheimer's disease. As a result, we wanted to conduct research that reflected our interests. This work may serve to improve new approaches that might help with drug development for not only Alzheimer's disease, but in the context of other neurodegenerative diseases. So next, we're going to be talking about the relevance behind our research. So Alzheimer's disease can be characterized as the cognitive impairment and progressive neurodegeneration. It is a disorder that involves loss of function and death of neurons, which is influenced by various proteins and pathways that can result in memory loss and regression in thinking ability. One of the underlying causes of Alzheimer's disease is the beta secretase abnormal cleavage of the amyloid precursor protein, or APP, located at the memory of the endosome, causing the amyloidogenic pathway that leads to the formation of A-beta plaques. Studies have shown that abnormal hyperphosphorylation of tau causes the protein to break away from the neurofibrillary tangles and form tangles in the brains of Alzheimer's patients. However, the effects of mutant APP and tau fly lines leading to the aggregation of A-beta and tau phosphorylation is uncertain, and further research is needed to address these pathogenesis of Alzheimer's disease. This proposal will use Drosophila to better understand the causes of neuronal loss caused by the overexpression of APP and tau, which is necessary for genetic screening.
We will be using Drosophila Melanogaster fruit flies for our experiments. The fruit fly has a fully sequenced genome and is used as a model organism for experimental studies in genetic manipulation, behavior, as well as anatomical studies. The animal model has a long history in research, which allowed for a multitude of well-established molecular genetic tools. Now, due to its short lifespan and cost-effective handling, this is an ideal organism for us to study our Alzheimer's research. Past studies have shown that the expression of disease-related gene products, like tau protein, causes distinct phenotypes, including irregular behavior and blindness in flies, which we hope to prove in our research as well. It mimics the Alzheimer's disease pathology through excess tau protein and APP production. Fruit flies have also been shown to be a prime model organism for the study of aggression to model behavioral components of the flies, which we will be using flight chambers to show this.
In order to understand the causations of Alzheimer's disease more in depth, we came up with 4 main aspects that we wanted to examine in our project. The first aspect would be examining the physical distribution of APP and tau in fly brains. Levels of APP and tau and normal expression of those proteins. And our second aim would be understanding more about how these proteins, APP and tau, affect fly behavior, specifically in aggression. And our third aim would be using the results from aim 1 and aim 2 to kind of infer the functional roles of different brain areas. And our aim 4 would be focusing on the role of sleep deprivation on APP and tau levels. For our approach and methods, we would be using 3 preliminary studies, GAL4 and UAS system, immunocytochemistry, and 5 chambers.
So the UAS GAL4 expression system is largely used to express endogenous and exogenous sequences in the tissue of interest. In this method, the driver line carries a GAL4 coding sequence, which is inserted downstream to an endogenous Drosophila gene. The main effect of GAL4 is to bind to an upstream activating sequence, or UAS region, that activates gene transcription to express the desired protein. By utilizing the UAS GAL4 system, Drosophila can be used as an Alzheimer's disease model by expressing the associated genes APPi and Tau in a wild-type fly background. The system develops an offspring of Drosophila lines that can express human protein in the desired tissues. The technique has been developed to disrupt the activity of single genes by expressing a transgenic RNA interference construct into the genome. Under UAS control, RNAi lines will briefly express in very few repeated sequences, which correspond to endogenous gene. The RNAi transgene techniques make studies of the targeted nervous system more attainable.
Our second way of studying our research was immunocytochemistry. We use ImageJ as a semi-automated approach to analyze a cellular process or to quantify protein aggregation in the Drosophila brain with fluorescent tagged mutant proteins. The immunohistostaining of brains of our fruit flies with APP and Tau will allow ImageJ to annotate patterns of gene expression. The progressive spreading of the green fluorescent protein labeled neurons within the brain is revealed by immunocytochemistry between the control groups and the APP and Tau expressing groups.
Our third method would be using the 5 chambers to analyze the aggressive behaviors of fruit flies. So 5 scoring is a technique used by Dr. Krovis' group in their paper studying social behaviors of fruit flies. And similarly, we would be using this technique in our project. This is highly effective in objectively quantifying the aggressive behaviors. The natural resources that we would be using for the male fighting chamber is a headless female thorax and abdomen body, while the female fighting chamber, we will be using yeast paste. The next slide shows our chart that we will be using to score while we score the fruit fly behaviors. Some of the aspects that we will be looking at are chasing, headbutt, wing flick, wing threat, fencing, tussle, and boxing.
So next we will be talking about the results of our research. This is a source we found incorporated between these 2 groups that correlate towards Tau lines. We'll be referencing the study to our aim results 1 and 2. The groups below right here, we'll be using in our research is boxed. So for our results on aim 1, which focuses on the structural changes, experimental results show that the Tau expressing flies abolishes vascular pathology in the brain and leaving holes in the structure, while the Tau-free fly brain remains complete. The structural changes also occurred in the retina. The Tau expressing flies develop a rough eye phenotype and reduced eye size, while the Tau-free fly retina remains healthy. These results suggest the potential Tau and APPi cause structural degeneration we could examine.
Here are the results for aim 2, which we focus on behavioral differences. Since we were not able to conduct our experiment, this table shows the behavioral scoring we predict we would see based off of our research. We hypothesized that the aggression levels in flies expressing APP and Tau would be higher than those that were not. With the previous slides data, we created a bar graph of our behavioral scoring. It shows the behavior of the APP and Tau expressing flies had more aggressive behavior overall. Results in the paper suggest that climbing activities for tau-expressing flies is lower than tau-free flies. This may suggest lower aggressive behavior for our tau or APP-expressing flies as well. However, this potential result contradicts our hypothesis that tau or APP increases aggression. We would have to look into whether the climbing behaviors correlates to what we defined as aggressive behaviors, although we would have to see if the different pathways were different, or if we could put them together as aggressive and climbing as one.
For our results for AIM 3, in order to map the social with changes, we plan on using image J if we actually have our experimental data. But since we are not in the lab, we analyze our results from previous studies in AIMS 1 and 2. As we see in AIM 1, tau creates the vacuoles in the fly brains. The vacuolar tauopathy effect has been studied recently by Edward Lee's group at UPenn School of Medicine, and they found out that the vacuolar tauopathy is a new and rare form of dementia that actually affects the behavioral, language, and executive problems in flies. This explains how the structural changes in the fly brains causes a decrease in flies' climbing ability, as we saw in the results from AIM 2. As for the effects of sleep deprivation have on the levels of tau and APP, past studies papers have shown that human APP and tau disrupt sleep and produce memory impairments. Others have also found that sleep deprivation increases the tau levels. These evidence do suggest that tau and APP is very closely related to sleep, either by altering the fly's circadian activity or changing the tau levels, as well as affecting memory. However, as these evidence show that sleep deprivation increases the tau levels, but they have not yet shown that sleep could actually reduce the protein levels. Thus, we really hope to further conduct our experiments when we get back on campus in order to understand more about how sleep could potentially reduce the protein levels or not.
So in conclusion, our research is developed around the physical effects and behavioral effects. We also research about the vacuolization that led to lower activity level, and sleep deprivation affects memory and increases APP or tau levels.
For future goals, we first want to preface that we would like to do this experiment fully in lab when we are able to. And when we found information on the climbing behaviors correlating to aggressive behaviors, we want to know if they are the same pathway or are they not correlated at all. And we also learned that sleep deprivation causes an increase in APP and tau, but would sleeping actually decrease APP or tau? And here's our bibliography with the works cited.
And that is it. Thank you.
I hope you guys enjoyed our experiments.